welcome to Science with Tal. In today's video, we will cover the gustatory system and understand the physiology that creates the sense of taste. As a quick aside, the gustatory system is part of the chemical senses along with the olfactory system, which gives rise to the sense of smell. If you want to know more about the olfactory system, there is a video available on my channel. Now, this video on taste will be segmented in a pretty straightforward fashion. We will start by discussing the relevant anatomy of the gustatory system. Then we'll go over the transduction mechanisms that convert taste qualities to neural signals, and we'll finish with the neural pathways in the brain that give rise to the taste sensation. This map that we will cover will be available in PDF format in the description, along with the olfactory one. So, as you might expect, the main organ in our body that initiates the perception of taste is the mouth. More specifically, the first step in taste perception begins at the taste bud, which looks something like this. The taste buds are found mainly on the tongue, in structures called papillae, but they can also be found on the palate, the pharynx, the epiglottis, and upper third of the esophagus. When we consider the papillae of the tongue, which again are the main sites where we can find taste buds, there are actually 4 different types of papillae that vary in shape, function, and location. The interior 2/3 of the tongue are composed of fungiform papillae, whereas the posterior third are composed of circumvallate papillae. On the posterior sides of the tongue, we can find the foliate papillae, and as you can see, in all 3 of these structures, we can find taste buds. There is also a 4th type of papillae, which is called the filiform papillae, and this one serves to provide grip to the surface of the tongue only. As such, it has no taste buds. Only the circumvallate, foliate, and fungiform papillae have taste buds. Alright, now that we know where to find our papillae and most importantly our taste buds, let's discuss the cellular composition of these buds. So broadly speaking, taste buds are clusters of about 50 to 100 taste receptor cells with some supporting cells and basal stem cells. The taste receptor cells, as you can see here in light violet, are responsible for transducing taste hints into neural signals. The neural signals produced by the taste receptor cells are then picked up by the afferent fibers and sent to the central nervous system for processing. Once we eat food, the taste hints inside of it get dissolved in saliva and come into contact with taste receptor cells via the gustatory or taste pore. This small opening allows to concentrate taste hints with the small hair-like protrusions of taste receptor cells called microvilli. The microvilli are structures that contain the receptors performing the transduction step, but we'll get back to this entire process in more detail shortly. Now, given their exposure to the outside world, taste receptor cells have a short lifespan that ranges from days to weeks. Accordingly, this turnover is balanced by the basal stem cells that provide a pool of future taste cells. The taste receptor cells, along with the supporting cells, can be further classified as type 1, type 2, and type 3 cells. Type 1 cells correspond to the supporting cells, and as their name says, their function is to support the taste receptor cells as they seem to have some glia-like properties. The exact details of their role is not fully understood yet. For type 2 and 3, those are both divisions of taste receptor cells. As we will see in a bit, there are 5 main taste qualities that we can sense: sweet, umami, bitter, sour, and salty. Type 2 cells, here in blue, are responsible for sensing the first 3 types, so sweet, umami, and bitter, and type 3 cells, on the other hand, in green, sense the sour taste. Unfortunately, it is unclear as of yet which type senses the salty taste. With this in mind, however, we have what we need to discuss the transduction pathway for each taste hint. We will begin by covering the mechanisms in type 2 taste receptor cells. Now, I should begin by saying that the field of transduction in taste buds is one that keeps on growing year after year, and this can be seen from the amount of new discoveries made in recent years. Accordingly, some of the details that I will present right now might diverge from your textbook or class notes. It is entirely possible. Even in my own readings to make this video, some of the reviews I took contradicted the typical textbooks I use. As such, if you have any questions or you want to know where I took some of the information, do not hesitate to ask me in the comments and I'll redirect you to the appropriate resource. So, moving forward, as we said, type 2 receptor cells detect sweet, umami, and bitter. The main reason why these 3 tastes are lumped together is because their transduction process is all mediated by a G-protein coupled receptor, or simply GPCR. The other tastes, so salty and sour, use different mechanisms that we'll see later. So, if we consider sweet first, the taste is detected by a heterodimeric GPCR made of a T1R2 and T1R3 subunit. Common chemicals that will elicit a sweet taste include sugars, artificial sweeteners, some proteins, and amino acids. For the umami taste, it is detected by a similar receptor to sweet, but instead of the T1R2 subunit, it has a T1R1 subunit paired with a T1R3 subunit, which again forms a heterodimer. The umami taste is associated with the savory taste of amino acids like aspartate and glutamate. The typical example that is used for umami is monosodium glutamate, and common foods that will elicit this taste include meats, broths, and mushrooms to name a few. You will notice that for both sweet and umami, I have written and more after the receptors. This is because while these are the best described mechanisms, there seems to be other pathways for the cells to sense sweet and umami that are independent of T1R receptors. For our purposes, we'll still just focus on what is better known. Now, in contrast to sweet and umami, the bitter taste is detected by a monomeric GPCR of the T2R family. In humans, there are more than 25 genes associated with the T2R receptors, and they all code for receptors capable of detecting bitter tastes from foods like caffeine, nicotine, alkaloids, and many more. Because of this wide variety of receptors, they all have different properties. Some can detect only one type of compound, while others can detect many different compounds. One common trait with all the bitter receptors is that they generally have a lower activation threshold. This is super important because the bitter taste is typically aversive and warns against the ingestion of toxic chemicals. As such, a low threshold and all the overlapping of detection across the T2R genes allows us to be well equipped to detect potentially noxious chemicals. This is in contrast to the sweet and umami tastes that are typically seen as appetitive, and they invite the consumption of rich foods. As such, the threshold can be a little higher. Alright, now that we have a rough idea of how these three tastes are detected, let's jump into the transduction pathway, which in this case is how chemicals are converted into a neural signal. An important detail about type 2 taste receptor cells is that although they include the bitter, sweet, and umami tastes, different type 2 receptor cells have non-overlapping expressions of T1Rs and T2Rs. In other words, a given taste receptor cell will either detect sweet, umami, or the bitter taste. That line is sometimes a bit blurred between sweet and umami, but never a taste receptor cell that detects bitter will also detect sweet or umami. Fortunately for us, all three tastes share a common downstream pathway, so we only have to cover it once to understand it for all three tastes. Because of what I just mentioned about the expression of the receptors, the starting point is either the T1R receptors, so sweet or umami, or the T2R receptors that detect the bitter taste. Now, when the respective taste tint activates the GPCR, this activation causes the release of the beta-gamma subunit that then activates phospholipase C beta-2, or simply PLC beta-2. If you need a reminder on GPCRs and second messengers, make sure to check this video where they are covered more thoroughly. Anyhow, the active PLC beta-2 then cleaves PIP2 to make the second messenger DAG and IP3, and the one that's interesting to us is IP3, as it opens a calcium receptor on the endoplasmic reticulum to liberate some calcium in the cytoplasm. This calcium allows to open a calcium-gated cation channel by the name of TRPM5. Upon opening, TRPM5 brings sodium inside the cell, and together with calcium, the ions depolarize the taste cell until an action potential is triggered. As a quick note, although taste receptor cells are electrically excitable and can generate action potentials, they are not neurons but rather specialized epithelium. Now, this action potential contributes to the release of ATP through a large pore membrane channel called CAH-LM13. The ATP released by type 2 receptor cells can then do many things. Of importance for us, it opens its related ionotropic receptor, the P2X receptors, on the afferent terminals to depolarize them and transmit the taste signal to the central nervous system. Additionally, and we'll have a word on this later, ATP is important for some autocrine and paracrine communication pathways between type 2 and type 3 taste cells. Finally, the ATP signal is terminated by degradation from type 1 cells by mechanisms that are not shown here. As a few quick notes about this pathway, type 2 cells contain a typical mitochondria and as you can imagine, these organelles are crucial for the proper production of ATP and thus the communication with afferents. Additionally, you will notice that the type 2 taste receptor cell does not release its contents in the conventional way that neurons do. Instead, they use what is referred to as a channel synapse, which simply means that they release their contents via this large pore channel. You will see later that this mechanism is different from type 3 taste receptor cells. Finally, although ATP is the principal neurotransmitter attributed to type 2 taste receptor cells, these cells can also release acetylcholine and a variety of hormones that further modulate the signal all around the taste bud. However, these details go a bit outside the scope of this video. With this being said, let's move on to the type 3 taste cells, which transduce the sour taste. In contrast to what we just covered, the sour taste is not detected by GPCRs, but rather by intracellular acidification. The compounds that elicit an acid taste are mostly acids such as citric, acetic, or malic acid, and many more exist. Type 3 taste receptor cells can get acidified by two main mechanisms. The first is via a proton channel called autopetrin 1, or simply OTOP1, and the second is via the ability of some weak acids to permeate through the membrane. In any case, the entry of these acids depolarize the cell because more positive charges are inside, and it also acidifies the cytoplasm by decreasing the pH. The decrease in intracellular pH blocks a particular leak potassium channel called KER 2.1. Because of this, potassium stays trapped in the cell and further depolarizes it. Together with the depolarizing effects of the added protons, the net depolarization generates an action potential, which is then transmitted throughout the cell to open voltage-gated calcium channels. The calcium that enters from these channels then serves to cause the exocytosis or release of serotonin, also known as 5-HT, into the synaptic cleft. The serotonin binds to its cognate ionotropic receptors on the afferent terminals, and this allows to depolarize them to send a signal to the central nervous system. As you can see, type 3 taste receptor cells have a more conventional synapse. One thing about this process is that the main neurotransmitter of type 2 taste receptor cells has not been established yet. Along with serotonin, type 3 taste receptor cells also release GABA and norepinephrine. Anyhow, this should get us a pretty good idea on how the sour taste is transduced. Finally, we arrive at the last taste, salty. Unfortunately, as I said previously, we actually do not know much about the taste receptor cell that transduces this sensation, nor do we know much about the downstream mechanisms. What is known is that in rodents, salt is detected via an ion channel called the epithelium sodium channel, or simply ENaC, but we do not have sufficient proof yet to claim that it operates the same way in humans. It will be interesting to see in the next couple of years if the scientists working on that can manage to find the answer to this enigma. Before we move on to our next section, I want to briefly mention some topics that we have not explored in our discussion, but are certainly of interest. First off, in addition to the neurotransmitters that we have mentioned, like ATP, serotonin, GABA, acetylcholine, and norepinephrine, it turns out that taste receptor cells also synthesize and release hormones like GLP-1, NPY, CCK, ghrelin, leptin, and many more. If you are familiar with the digestive system, you might recognize some of these hormone names as they are heavily influential there. Given the close relationship between what's going on in the mouth and the digestive system, it's intuitive to imagine that there is a hormonal axis between the two, and having this additional layer of regulation allows to fine-tune the sensitivity of taste receptor cells and even on our own feeding behaviors. If you are interested in this topic, I suggest going over this review of the endocrinology of taste receptors. It is truly fascinating and goes over some niche topics about the gustatory system. Anyhow, the second topic that we briefly mentioned is the presence of numerous paracrine and autocrine regulatory mechanisms. For example, type 2 taste receptor cells, which release both ATP and acetylcholine, have receptors for both molecules on them and thus can influence their own activity in an autocrine fashion. As a quick example of a paracrine mechanism, type 3 taste receptor cells also have ATP receptors and thus get modulated when type 2 receptor cells release ATP. If you imagine that these mechanisms also exist for the hormones that we just mentioned, the signaling in the taste bud can get pretty extensive. Additionally, some studies have reported that taste receptor cells might communicate between each other through electrical synapse via gap junctions, and again, complexifying the signaling in this region. Finally, the 5 tastes that we recognize are not set in stone. There is some literature to support fat to be a taste, but this concept remains debated. Again, it will be interesting to see in a couple of years what gets discovered and how it changes our approach to the taste bud. Alright, going back to this figure of the taste bud, we have a good idea of how type 2 and type 3 cells transduce tastins into neural signals. We also know that it seems that taste receptor cells are sort of separated in a discrete fashion, with some taste receptor cells only detecting sweet, others detecting umami, others detecting bitter, others detecting sour, and presumably others detecting salty. This segregation of signals leads to the idea of label line coding, or in other words, the concept that each taste cell is responsive to a given taste, and the signal of that cell is conveyed to the central nervous system by an afferent of the same taste quality. So sweet-detecting cells send their information to sweet-detecting afferents, bitter cells to bitter afferents, and so on. While there is a lot of evidence to support this model, and it is likely that your resources mention label line coding as the way the taste bud operates, it doesn't explain everything, and the actual mechanism is likely to be a bit more complicated, namely because of all the paracrine and autocrine mechanisms in the taste bud. But whether label line coding is the main model or not, let's go back to the diagram of the tongue to discuss what the afferents are and where they are carrying the signal to. So recall that we can separate the tongue in the anterior 2/3 and in the posterior 1/3. The anterior 2/3 contain mostly fungiform papillae, whereas the posterior third contains circumvallate papillae. It turns out that both of these regions are innervated by a different cranial nerve, and this is what constitutes the afferents. So, for the taste sensations in the anterior 2/3, the facial nerve, or cranial nerve 7, is responsible for picking that up via the chorda tympani branch. The taste in the posterior third is picked up by the glossopharyngeal nerve, or cranial nerve 9. As you can see, there is a small overlapping portion at the level of the foliate papillae, which gets innervated by both cranial nerve 7 and 9. Lastly, the last cranial nerve to pick up taste is the vagus nerve, or cranial nerve 10, and it does so from the pharynx, epiglottis, and soft palate. All 3 of these cranial nerves send their afferent information to the nucleus solitarius, which we will see in more detail shortly. As a final note, I want to mention that the cell bodies of all 3 cranial nerves are located in specific ganglions of the face, namely the nodose, petrosal, and geniculate ganglions. To illustrate a little better the gustatory system pathway, let's consider a sagittal cut of the face, which will allow us to better see most of the relevant structures. So, as we said, beyond the taste buds, the first step in the pathway is for the nucleus solitarius to receive afferent information, and there are 3 cranial nerves that bring taste information to the brain. The facial nerve, or cranial nerve 7, which brings taste from the anterior 2/3 of the tongue, the glossopharyngeal nerve, or cranial nerve 9, that brings taste from the posterior third of the tongue, and finally, the vagus nerve, or cranial nerve 10, that brings taste information from all the other areas around the oral cavity. All these taste fibers are brought to the nucleus solitarius, or nucleus of the solitary tract, in the medulla division of the brainstem. To be more precise, the taste information gets received in the rostral nucleus solitarius, which is also known as the gustatory nucleus. Now, to find this gustatory nucleus, let's take a look at the interior surface of the brainstem. In other words, we'll look at the brainstem from this direction. The extent of the nucleus solitarius, as highlighted in light red, is pretty long and is generally divided into a rostral and caudal section. To put it simply, rostral is towards the head, and caudal is towards the feet. So if we take a cross-section at the rostral medulla, this is where we'll be able to find the rostral nucleus solitarius, or simply the gustatory nucleus. For your information, the caudal part of the nucleus solitarius deals with various visceral sensations and homeostatic reflexes of the cardiovascular, respiratory, and gastrointestinal systems, so it's a pretty important area that receives a ton of connections from different systems. Now, with respect to our gustatory pathway, the taste information in the gustatory nucleus is then sent to the ventral posterior medial, or VPM nucleus of the thalamus. The VPM is also responsible for receiving somatic information from the face and oral cavity, which include sensations about touch, pressure, pain, proprioception, and temperature. After thalamic processing, the gustatory information in the VPM is then sent to the gustatory cortex, where the conscious perception and discrimination of taste stimuli will occur. This gustatory cortex is located in the anterior insula and frontal operculum portion of the brain. To find this area, we need to consider a lateral view of the brain, in which you will see the lateral or sylvian fissure here highlighted in blue. If we open up this fissure, this will reveal the insula, which is simply a portion of cortex that is typically hidden when we look at it from outside. The cortex that overlays the insula is called the operculum, and there is an operculum in the frontal, parietal, and temporal lobes. The one that interests us is the frontal operculum in red, so if we take a coronal cross-section at that level, the region highlighted in the figure represents the gustatory cortex. Now, one important point that I would like to briefly touch on is the concept of flavor, which is different from taste. As we've seen, taste represents the basic sensations of sweet, bitter, umami, sour, and salty. On the other hand, flavor is created from the multisensory integration of gustatory, olfactory, and somatosensory systems. This flavor is what you experience every time you eat something, rather than the pure taste detected by your tongue. This is an important distinction to make, and it explains the common saying of flavor or taste is 80% smell. Indeed, when you eat, volatile molecules from your food go back to the nasopharynx, where they'll interact with the olfactory system, and this will produce the unique flavor we experience. While still important, the tongue only detects the 5 tastes that we have mentioned and doesn't discern between, let's say, an apple flavor versus an orange flavor. It will only be able to tell whether it's sweet, sour, bitter, umami, or salty. This relationship between taste and olfaction explains why food can taste so bland when you are sick and your nose is stuffed. Finally, I want to mention that the gustatory cortex makes an important connection with the hypothalamus, which, among its many functions, is responsible for feeding behavior and food-related autonomic behavior. The subject of feeding behavior is really complex because it pulls information from energy homeostasis, or in other words, your energy levels, so whether you're hungry or not, but it also pulls information from your cognitive states, so for example, your mood, emotions, your memories, your conditioning, and it puts it all together along with some other signals to control how you behave around food. So as you can imagine, our taste sensation plays an important role in this, and thus it is intuitive that the two connect together. If you are interested in this topic, I suggest consulting this review that discusses the pleasantness of food. It summarizes some of the key areas and research findings that have allowed us to infer how feeding behavior might be orchestrated. Alright, with this being said, this concludes our discussion on the gustatory system and overall on the chemical senses that also included the olfactory system. If you want to consult these maps, they will be in the description of the video. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.